I'll just be quick because we'll just a touch behind. So it's our pleasure this week to welcome as our colloquium speaker Dr. Bärbel Hönisch from Lamont-Doherty at Columbia University. So Dr. Hönisch is I think really pioneered the use of boron among many other things that she's pioneered to understand the past of Earth's ocean and climate system. And today she's going to be talking about reconstructions of Cenozoic atmospheric CO2 and ocean carbon cycle perturbations. So thank you again for making the time to be with us. We're really excited to have you.
Absolutely. I'm very excited to be here. Thank you so much for having me. Haven't met with very many people yet just because I just arrived, but I'm excited to be here. And I wanted to tell you a little bit about some work that I've done over the last couple of years. So yes, I have worked on boron isotopes in my life. You'll get to see a little bit of that. But I mostly want to, and I assume that's why I mostly got the invitation, was because we recently published a paper on paleo CO2 over the Cenozoic. So we'll get into that in the end as well. But so there are a lot of people that I have to acknowledge on this. They're basically all in this paleo CO2 logo up there. That's actually our website. If you happen to be interested in that kind of thing, there are a lot of FAQs and information about proxies and all kinds of things. Now, how do I move? Let's just go that way. No. Okay, cool. So before I go in telling you about any research, I figured I'll tell you of who I am and where I came from, and it's sort of a meandering path. So when I first started out, at school I took two majors, and one of them was art and the other one was biology. And so in my first life, I thought I wanted to restore paintings. That turned out to be a rather difficult path, and that didn't work out. I'm still sad about that. But then I thought, okay, so if I don't get to do anything with paintings, then maybe I'll go into biology. And I flipped through an encyclopedia, and I found the list of the Max Planck Institutes in Germany. And there was one on marine microbiology. And I kind of skipped the micro, and I thought marine biology sounds great. There are going to be dolphins and whales in my future. As it turns out, there are not very many people who work on dolphins and whales. So I got to work on plankton pretty quickly. So I went to Bremen. That's why I studied mostly biological oceanography at first, learned about all this plankton stuff. And then I ended up doing my diploma thesis actually in the geology department, where I worked on otolith. So they had found all these otolith up and down the coast of Namibia in sediments. And at first they didn't even know what they were. And then they thought, well, maybe biologists can find out who they belong to. And that was kind of fun. So that was my first experience with isotopes and other things. And then I did my PhD with Jelle Bijmer at the Adric Wegener Institute. And Jelle is somebody who has kind of pioneered culturing planktic foraminifera for calibrating paleoproxies. So that's a picture of a living foram up there in a culture jar, that tiny little speck, if you happen to see it. Now this is mostly how we get to see foraminifera. Later on, I moved on for a postdoc at Lamont, actually. That was my first experience not exactly my first experience in the US, but my first research experience, where I finally went down core and did some paleo reconstructions. And now the last thing that I've been spending the last 7 years of my life on was mostly this paleo CO2 network. I'll show you some of that later on of what we were doing there. So before we get there, I wanted to go back a little bit and talk more about carbon cycle perturbations before we go to the CO2 in itself. And so this is a figure that looks at 70 million years of climate change in wiggly lines, as we like to look at in paleoceanography. On the top, you see the benthic isotope stack, and on the bottom, there's a carbon benthic oxygen isotope stack, benthic carbon isotope stack at the bottom. And you see all these ups and downs, and they tell us about all kinds of different things. In particular, what's most interesting is that these oxygen isotopes here have gone heavier and heavier, so they're getting more and more positive, and that's an overall cooling trend over the last 70 million years. There's some ups and downs in there. 34 million years ago, somewhere over here, if you look up there, that's the transition from a green Antarctic continent to an ice-covered Antarctic continent. You see these big changes in the isotopes there. This is a really interesting event, the Paleocene-Eocene thermal maximum we'll get to talk about a little bit later on, where we have a massive warming event and some really interesting carbon cycle perturbation. And then in the end we have these big ice sheets that we have in the ice ages over the last couple hundred thousands of years or couple millions of years. So of course it's sort of really interesting to find out what was happening at these different times. Why did the climate change? How did it change? What did carbonate chemistry do at that time? The reason why we're interested in carbonate chemistry is, of course, because right now we're burning a lot of carbon dioxide and we feel that the Earth is getting warmer. And so that's, on the one hand, that's the greenhouse effect, and those are these two parameters that I'm interested in my research. On the one hand, we have more carbon dioxide in the atmosphere that makes the planet warmer. So that's the global warming part of this. The other one is that you actually, the carbon dioxide also dissolves in seawater, and once it dissolves with seawater, it forms an acid, and that's carbonic acid, and so that leads to ocean acidification. And so those were the two parameters that I learned about when I was a graduate student. Ocean acidification, I was probably involved in one of the first ocean acidification experiments on coccolithophores as an undergrad, where I was doing these experiments in the lab, and that's what kind of motivated me to move forward and do more of this research on my own to contribute to this understanding. So this figure here, so the ocean acidification part there is interesting because it affects marine calcifiers. So these are pictures of an experiment that I haven't done myself. That was published elsewhere, where they cultured pteropods, planktic snails that are made out of aragonite, in the laboratory under various different pH conditions. And the ones on the right-hand side, this beautiful translucent shell, that's under normal conditions as we still see them today, even though that's already somewhat acidified. And the one on the far left side, that's a shell that has lived for a little over 40 days in conditions that we expect to see by the end of the century. And you can see that the shell is malformed, it's getting thinner, it provides less and less protection for the organism that's in it. And so marine calcifiers are some of the main organisms that we're interested in, of what is this going to do to in the future as we produce more and more carbon dioxide. Now, the global warming signal we can see very, very clearly over the last couple of years. Here's a timeline from 1880 to 2020. That's a temperature anomaly here, so it looks at the temperature change relative to the 1950s. It was a little cooler before then and a bit warmer afterwards. It keeps getting warmer. The last year was just the hottest year on record, again, which seems to be happening about every single year these days. On top of that wiggly line, those are the carbon dioxide measurements that have been collected at the Mauna Loa Observatory since 1958. And so you see a pretty nice correlation between carbon dioxide going up and temperature going up. And we've known this for a very long time, that carbon dioxide is a greenhouse gas. Now, of course, we want to go back in time and we want to know how did this happen in the past? Is this something that only happens today, or is this something that has been around for a very long time? And then when was the last time when carbon dioxide was as high as it is today and as it might be by the end of the century, and what was the climate like at that time? So what can we learn from that going back in time? But these actual measurements, they only go back to 1958. If we want to learn more about carbon dioxide, then the first thing that we can do is we can look into ice cores. So ice cores are really cool because as the snow is falling on Antarctica, once that snow gets thicker and thicker and gets more and more compact, it actually encloses bubbles of ancient air into that ice. And that's one thing that you can see in this picture over here. So this is a slice of ice from one of these ice cores, and all these little dots in there, those are bubbles of ancient air. And so what the scientists are doing, they just slice up these ice cores and they melt them, they let them melt, and they extract the gas that's coming out of that, and they can analyze this. And this figure over here, that's it only goes back to 800,000 years so far in carbon dioxide. So the bottom part shows the carbon dioxide, the top shows the temperature estimates from the deuterium isotopes that were measured directly in the ice. And so you can see with the deuterium isotopes, that's a temperature indicator. And so you can see how the temperature is going up and down over time. About once every 100,000 years or so. And whenever the temperature is high, the carbon dioxide is high. And when the temperature is low, the carbon dioxide is low. So that's a pretty nice relationship that tells us, again, that carbon dioxide and temperature are very tightly correlated. Here on the right-hand side, on that yellow bar, that's just the Mauna Loa record. Very different time scales. These are 800,000 years. That's 1958 to 2023. I haven't updated that this year yet. Now the ice cores, they're out there drilling right now. There's several groups that are in Antarctica, and they're trying to take more ice cores, and they're trying to go back to about 1.5 million years if they find that ice, if they're lucky with that. And that's really cool, because these are just really, this is sort of the gold standard of carbon dioxide that we can find in the past. There's nothing better than measuring the actual thing. The problem is that the ice, the more ice you load on top of the continent, at some point it starts melting at the bottom, and it actually also moves. It slides off the continent, and so we don't have endless years of ice that go back to 34 million years ago, when Antarctica actually started glaciating. And so in order to find out what the carbon dioxide concentrations were further back in time, we have to go and look for proxies. And so the proxy that I'm using is the boron isotope proxy that specifically looks at the acidification part of that of the record. So here on the upper right-hand side, you see two figures. The top one shows you the concentration of the two dissolved species of boron in seawater. That's boric acid in red and borate ion in blue. And they're shown relative to seawater pH. And what you can see is that at low pH, pH 7, pretty much all boron in the ocean is present in the form of boric acid. And then at higher pH, that boric acid dissociates to borate ion and hydrogen ions. And so at high pH, boron is present in the form of borate ion. Now, the lower figure here, that shows you the isotopic composition of these two species of boron. So there are two isotopes of boron, two natural isotopes of boron, that's boron 10 and boron 11. And because of differences in how tightly they're bound in these different boron species, boron molecules, boron 11 prefers to be in the form of boric acid, and boron 10 prefers to be in the form of borate ion. So if you look at this figure here, so this is again the boron isotopic composition over seawater pH. The stippled line over there, that's the isotopic composition of boron in seawater. That's just the dissolved boron, so that is constant at 39.6 per mil right now. And because all boron is present in the form of boric acid at the low pH over here, that means that the isotopic composition of boric acid must be approximately 40 per mil at low pH. At high pH, the isotopic composition of borate ion must be close to 40 per mil. Between these two, there's an approximately 27 per mil fractionation between these two boron species, and that's what's shown in this epsilon value over here. Now, my postdoc advisor at the time is Gary Hemming, and he, in an earlier study, measured boron isotopes in a range of different marine carbonates. So that's this box over here. He looked into corals, he looked into ooids, into foraminifera, into sea urchins, and all kinds of things. And it turned out that those carbonates all had an isotopic composition that fell sort of in this box over here, which is an indication that it's the borate ion that is incorporated into these marine carbonates. Now, if that were true, then you would expect that the isotopic composition of these carbonates should increase as the pH increases. But this is something that's really difficult to test in the ocean, because if you look into the ocean today, you have a pH range that is sort of 8.15 plus minus 0.1. So it's a very, very small pH range that you see in the surface ocean today, and that doesn't really allow you to calibrate these proxies in a very significant way. And so that's where my experience with culture experiments came to help again. So I've been doing culture experiments with planktic foraminifera since my PhD. Up there on the right-hand side, that's this little speck over here with that little fuzz around. That is a single specimen of a planktic foraminifera. What we do is we take them from the ocean. We used to go diving for them. The last 2 summers that we were doing these experiments, they didn't allow us to go diving anymore because they thought it was too dangerous. But we take these juvenile foraminifera into the laboratory. We identify the species. We measure how large they are when they're coming in. And then we make our own seawater jars where we can change anything we want in that seawater. We can change the chemistry, we can change the pH, we can change the temperature. They're sitting in these temperature baths where we control the temperature and we feed them every other day. And so we can basically make that form live in any kind of ocean that we want it to live in. And then we can eventually, once they have completed their life cycle, which doesn't take very long, it's just a couple of days, then these specimens undergo gametogenesis. So they turn their entire cytoplasm into gametes. They release those gametes, and then the empty shell sinks to the bottom of the ocean. In our case, it sinks to the bottom of the jar, and that's where we collect it, and then we can analyze it. The problem why we're doing it in that elaborate way is because planktic foraminifera we still haven't figured out after decades of research of how to reproduce them in culture. So we can really only work with these organisms that have spent some of their life cycle in the ocean. But you can, now these are pictures of planktic foraminifera, so in some cases when you know how large they were when they came into culture, you can measure their size again when they have been cultured and when they've completed their life cycle. And then you can go with a scalpel under the microscope and chop off these little chambers that have been secreted in the lab that have seen just the conditions that we wanted to analyze. Tedious work, but it brings you to some calibrations. And here we go. Here's the boron isotopic composition relative to seawater pH of different species of foraminifera. The blue squares up there, that's actually a benthic species of foraminifera that Fiorella is working on. These three lines over here, the colored lines, those are all planktic foraminifera. And then the black line is actually a synthetic calcite that was also secreted in the laboratory. And what they all have in common is that you see that the isotopic composition increases at higher pH. And so that's all that we need in the end. So we have established this in the lab, but now we can go in there, we can take these shells from the sediment, measure the isotopic composition, and then read what the pH was at the time when these organisms were living. So during my postdoc, I then went on and tried to verify that this also works in the past. So here's a figure that looks at ice core CO2 in the black line over, well, when I first started, there were only records that went back to 450,000 years. So the black line is the ice core CO2, and then the blue symbols and the red symbols, those are reconstructed seawater acidities from planktic foraminifera. And you can see that it's actually working pretty well. So when the CO2 is low, the pH is high, and when the CO2 is high, the pH is low. And we get these actually really quite nice cyclical patterns that correlate with that pretty nicely. We can go further. So as you've seen before, the more acidic the seawater is, the higher is the CO2. So from there, we can actually estimate what the carbon dioxide concentrations were in the surface ocean, even though we have to be careful where we're going. But you see, overall, over glacial, interglacial timescales with these 100 or so ppm differences in CO2, we get a difference in seawater acidity on the order of 0.15 pH units. Now, you can look into the literature, and then you can find all kinds of information about what does this do to calcification. So here are two different studies. The one on the left-hand side, the time here goes from right to left. Right is old, left is young, just the last 50,000 years. And the black line there, looks like green over here, but I hope it looks black to you, are the oxygen isotopes, so it just tells you what the climate state is. And then the red line, those are normalized, this is normalized weights of planktic foraminifera. So you can see that the foraminifera during the last ice age, 20 to 30,000 years ago, when we know the pH was higher, they actually were heavier compared to what they are in the modern ocean. So there's a decrease in the calcification thickness of these foraminifera as the ocean was getting more acidic. That's for foraminifera. The right-hand side shows you studies of coccolithophores, so they're a form of calcifying algae. And in this case, time goes from left to right. So left is old, 40,000 year, right is young. Here's the last glacial maximum. And you can see that the weights of these individual coccoliths are also much heavier during the last glacial compared to what they are in the more recent times. So 0.15 pH units, they're already making a big difference in the calcification response of these organisms. So to sum this all up, just as a reference point that you should keep in mind is the carbon dioxide changes glacial, interglacial are on the order of 100 parts per million. We have about 2 to 3 degrees warming that is associated with that increase in CO2 as well. There's a decline in calcification as the water is getting more acidic. Took about 6,000 years to get there, to go from 280 ppm to 180 ppm. Actually, the other way around, 180 to 280. And so that means, and the pH decrease is 0.15 units. And so if you divide that by 6,200, then you come up with an acidification rate of 0.002 units per century. Now that's very different from what we're expecting by the end of this century. We're expecting to see a pH change on the order of 0.2 to 0.3 pH units. That just happened over the course of maybe 100 years or maybe 200 years. It's speeding up as we keep going. So, of course, we want to know, what does this do? Are there other times in Earth history where we can actually look at this? And the one time interval that is really, really interesting here is this so-called Paleocene-Eocene thermal maximum here about 56 million years ago, where the oxygen isotopes up there, they indicate that there was a massive warming event. And the carbon isotopes tell us that there was an input of some dormant carbon reservoir, whatever that may have been, whether it was methane, whether it was volcanism. We will get back to that later on of what this was. But the carbon isotopes tell us that there was a huge amount of carbon put into the system. Now, another indicator for acidification actually comes from observations of the seafloor sediments, where we can look at sediment cores off the coast of Namibia. So these sediment cores here, they were taken here off Walvis Ridge of a depth transect that was collected somewhere over here. And these different cores, they indicate water depth from which they were taken. These are modern water depths. 2,700 meters to 4,700 meters is the deepest one where it was collected today. In the past, I believe this one was 3,600 meters, and this was only 1,500 meters. So at the PETM, they were much, much shallower compared to where they are today. But what they all have in common is that you see these color changes in those cores. So they all start. So time goes from goes up. So this is old, and up is young. And in the oldest parts of these cores, you can see that the colors are much lighter, and then all of a sudden they turn dark brown, and then they slowly become lighter again. You can see here from these blue lines, this is the percent calcium carbonate that went from pretty high, from almost 100% to 0, and then slowly recovered again. So the dissolution of these calcium carbonates in itself is already an indication that there was a massive dissolution event happening as a function of carbon dioxide put into the system and acidification in the seawater. So these brown layers, this is actually clay deposition. So the clays that are usually diluted by all these plankton organisms that fall down, they were dominating that sediment, and so they turned that sediment brown. Now the core on the right-hand side is actually here from the equatorial Pacific at the time, and this is again a picture of the core. So you can see again it's light at the bottom, brown at the event at 56 million years ago or so, and then it goes up and it slowly gets a little brighter again. These pictures here, they are pictures of the plankton composition or the fossil composition before the event, during the event, and then after the event. And the magnification of these pictures is the same in all of these three different pictures. So you can see just by looking at the size that what's happening directly during the event, that they're very much smaller compared to what they were before and what they were after. Now you won't be able to tell the difference in the species. They all look like popcorn to you, I assume. But it also turns out that the species during the event are different from the ones before the event and then also after the event. So there were excursion taxa that were occurring. And some of these species that lived before, they actually went extinct during this event, and then the new species that evolved afterwards. So all of this taken together tells us that there is massive calcium carbonate dissolution. There was dwarfism of these organisms, so they did not enjoy where they were living. There are these excursion taxa and these extinction events. So there was a massive impact on the ecosystem at that time. Now we wanted to quantify what the acidification would have been at that time. And so we did a bunch of isotope and trace element analyses. This was actually something that Don Penman did during his PhD. And so you have 4 panels here. We're just going to look at the upper 3 so far. Time goes from left to right. So this is 50. This is actually in centimeters, but centimeters in this core actually happens to be equivalent to 1,000 years each centimeter. So it's about 50,000 years before the event, and then we're going about 150,000 years after the event. And the top figure indicates the carbon isotopes that were measured in different species of foraminifera. And you can see that big decrease in the carbon isotopic composition, which is an indication for an input of carbon at that time, and then the slow recovery. The second panel over there, that's magnesium calcium. Magnesium calcium is a temperature indicator, and when magnesium calcium goes up, that means that it got warmer. And so all of these species are saying that, okay, so as the carbon was put into the system, we also see warming that's happening. And then the third panel here, those are boron isotopes that are measured for dawn. And so you can see that decrease in the boron isotopic composition, and then followed by an increase or recovery from that change. Now it turns out that this decrease in the boron isotopic composition, that is equivalent of a pH decrease of about 0.3 pH units. So very similar to what we're expecting by the end of the century. And so that's what makes this event so particularly interesting as a possible future event, or future analog to what might be happening. So those are the first 3 panels. The last panel here was something that we were all very excited about at the time, and that is the boron calcium measurements. Boron isotopes to this day are quite difficult to do. They're very time consuming to do. And boron calcium can be measured much, much quicker. And so for a while there was this thought like, well, if we can measure boron calcium, we might even be able to pair it with the boron isotopes and reconstruct the entire carbon system. Now what you can see is that boron calcium here decreased across the event and increased, and that's sort of what we expected. But it turned out to be a little more complicated than we thought. So to go back and explain a little bit what the boron calcium proxy is doing, is we're kind of following the information that we have from the boron isotopes that tells us that it's the borate ion in seawater that is incorporated into these shells. That's what we just know from the isotopic composition. And we see that the borate ion concentration actually increases at higher seawater pH. So that was a thought that, well, so if borate ion increases and that's what's incorporated, then we should probably get more boron incorporated into the shells as the pH goes up. And we've done that in culture experiments, and we saw exactly just that. So this is the boron calcium ratio over seawater pH done by a number of different graduate students. Kat Allen was my first grad student. Michael Heenan was never my grad student, but he's worked on this as well. You can see that in these different species of foraminifera, we see that increase in the boron calcium ratio. And we had some of these data before we did that PETM study, and we applied it, and we were kind of confused by it. The problem is that if you apply these calibrations, if we apply these calibrations to the PETM, and we just assume, okay, so we have a 0.3 pH unit change in the boron isotopes. And then you can use these calibrations over here to predict what a 0.3 pH unit change should do to boron calcium. And those are these solid lines here, using all these different calibrations. But relative to that, in black are the symbols that are the actual measured boron calcium ratios from these foraminifera at the time. And that show a much, much larger change in boron calcium. At first we actually went the other way around. If we turn those boron calcium ratios using these calibrations into a pH or into borate ion concentrations, we actually got negative borate ion concentrations in seawater, which didn't work. So we thought, we don't understand something about this proxy. Or maybe it doesn't work, or we don't really know. But Kat Allen had actually done some experiments while we were testing this proxy, where we also changed the total amount of carbon. So to walk you through this, so this figure here on the left-hand side, that's the boron calcium ratio over the carbonate ion concentration in our culture water. The blue line, these are our first experiments that we did where we changed just the pH. So what we did is we took our seawater, we added some acid and base to it. If you add base to it, you bring up the pH. that means that you also bring up the carbonate ion concentration. So with higher pH, the carbonate ion concentration shifts to higher values. And this is just the pH experiment over here. And with higher pH, the boron ion concentration will also increase. But as a charged species, so if boron is the species that is incorporated, then boron does not actually substitute for calcium, it substitutes for carbonate ion in the shell. And so therefore we thought, well, what if there is some sort of a competition going on between carbonate ion incorporation and boron ion incorporation? So we did a second experiment where we kept the pH constant, so we did not allow boron ion to increase, but we actually added sodium bicarbonate to the seawater, and that means that we increase the carbonate ion concentration by keeping pH constant and by keeping the boron ion concentration constant. And these black lines here, these experiments, they were now done at under increasing DIC conditions where the boron ion is constant, and you can see that there's actually the opposite compared to what we see with the pH effects. And so that is an indication that indeed there is some form of a competition happening. Now Laura Haynes was a more recent graduate student, and she actually extended this further. And we now also started mimicking ancient seawater. So we changed the boron concentration, we changed the calcium magnesium concentration. So we tried to make seawater as it looked like 50 million years ago. And she did more of these DIC experiments, and all of them are showing the same thing. If you increase the DIC, then the boron calcium concentration goes down. Now this is interesting because at the PATM, we have this notion that DIC must have increased at that time. So if you look into normal or modern ocean acidification experiments, this is a Bjerrum plot over here. This shows you the concentration of dissolved species of carbon dioxide on the left-hand side, and bicarbonate ion and carbonate ion on the right-hand side, but then also boric acid, so borate ion and boric acid as a function of seawater pH. And so in a normal acidification experiment, you go from a higher pH to a somewhat lower pH, and that means that the borate ion concentration is decreasing, the carbonate ion concentration is decreasing, and the bicarbonate ion concentration is increasing. And under those conditions, you would get a small change in boron calcium. But now at the PTM, when we had this massive increase in DIC, the borate didn't change, but the bicarbonate and the carbonate ion concentration were massively increased. And so that means that you actually get a much larger change in the boron calcium ratio compared to what you would find in situations where you only acidify the ocean. And so this now allowed us to actually deconvolve the pH effect and the DIC effect for the PATM, and to use these data that Don Penman had collected during his PhD. So what you see here is in green, so time again goes from left to right. This is before the event, this is after the event. In green, you see the normalized boron calcium ratios to the pre-event conditions of that now extinct species. Using the calibrations that we have from culture, we could calculate the borate to DIC ratio of the seawater at that time, and that's what's shown in this figure over here. Now we can use the boron isotopes to tell us what the borate ion concentration is. And so now we have borate ion concentration from the boron isotopes, and we can deconvolve this part here into borate ion from boron isotopes and the DIC. And that's what you can see over here on this figure. So the DIC is shown here in purple. So that shows you the big input of dissolved inorganic carbon at the PATM. And then we had to do some additional corrections there, because some of that, when you get seafloor calcium carbonate dissolution, you really only want to look into what was put into the system in the first place and not actually include that dissolution as well. And so we came up to a pretty large increase of 14,900 petagrams of carbon across that event. As a disclaimer, you have to say that these uncertainties here are very, very large. But this was one of the first ways of how we could actually do this completely with data. So for such a large addition of carbon, you kind of have to move away from ideas that this could have been methane hydrates, for instance. So methane hydrates have an isotopic composition, carbon isotopic composition of about -50. So with something that has an isotopic composition of -50, you don't have to add much carbon to the system to change the isotopic composition of the seawater and the foraminifera. You need something that has a less negative carbon isotopic composition. Volcanism has -5 per mil. Organic matter is about minus 25. And so the ideas of where this might have been come from is mostly thermogenic methane at this point. So that's a picture from an independent study that looks at Greenland, and it looks at this North Atlantic igneous province where lava was coming up underneath Greenland and Iceland, and this lava was actually moving its way through very organic rich layers of sediment. And so as that lava was burning up that sediment, it was not only the -5 per mil of the lava itself, but the -25 per mil of the organic matter that was put into the system. And with that, you can sort of estimate that this is going in the right direction. So that was one way of trying to quantify this at the PATM. Now, what is really interesting here, and what is the most important, sorry, I left one form in there. The most important part of this study is to look at. So we understand now that the acidification at the time was similar to what we're doing today. But what's really important here is to look at the overall changes. So overall, the warming that has been estimated for the PATM from the low to the high latitudes is on the order of 5 to 9 degrees. So it's a pretty massive warming event. Acidification is similar to what we're expecting by the end of the century. We have seen ecosystem changes, the dwarfism, the extinction, the evolution of new species. The onset of the duration, so just for this step here to happen, somewhat took on the order of 3 to 5,000 years. And with that, you get a pH change on the order of 0.01 units per century. So that's still 10 times slower compared to what we're doing today. And then the really important time is for that system to recover took 150,000 years. So that's a really long time. If we think about what is our carbon dioxide doing for us today, where is this going to go? If we wait for natural processes to neutralize all that carbon dioxide, that is going, now for our species, that's going to be with us forever. So here's just a picture from a paper that we published with Jess Tierney, going back in time 300,000 years. The black line, those are the CO2 estimates from, or the CO2 measurements from the ice cores. It's going up already, so we're now at over 420 parts per million. The gray and the white line, those are the estimates from the IPCC report of where we might be heading over the next couple hundred years, depending on what the different emission scenarios are. So it could go way up and could go way up to much warmer temperatures. But then over here you can see hundreds, thousands of years in the future of how long it takes for Earth to naturally neutralize that carbon dioxide. Just as a reference point, the Neanderthals died out about 40,000 years ago. So this is something that will be with us for a very, very, very long time. So these are all the things that I wanted to say about these carbon cycle perturbations in the past. What I wanted to do now is to come back to this picture and to talk a little bit more about all these other events that are happening over these last 70 million years. So there are lots of ups and downs and short increases and short decreases. And of course, we want to know what exactly was happening over all this time with the carbon dioxide. And we want to find out when was the last time that CO2 was as high as by the end of the century, and what was the climate like at that time. And so my colleague Dana Royer has done this for many, many years. He's actually spent most of his career just compiling all kinds of paleo CO2 estimates from all the different studies that have been published. And he looked into marine proxies, into terrestrial proxies, whether they're paleosalts, whether they're leaf proxies, liverworts, boron isotopes, or alkanones. He has compiled all these records, and he's kind of gone through the literature, and if somebody said, oh, this record is not good because of that, then he sorted them out. But he's not exactly an expert on every single one of these records. And so this is just one portion of a paper that was published with Gavin Foster in 2017 that used Dana's compilation. And you can see there's quite a lot of variability in these data between the different proxies, but also within the individual proxies. And so at that time, we started a research coordination network that was trying to work on this specifically, to try and bring the community together, all these different experts of all these different proxies that really knew what the current understanding of the proxies are, where the pitfalls are, and where we could really still trust some of the older published records or maybe could not trust them. And so here on the left-hand side, you see all the different proxy systems that we have looked at. There are these two marine systems. These are coccolithophores. They actually, it's not the outside of the shell that we're interested in. We're actually interested in the alkanones that they produce, and so that's in the organic matter of that shell that tells us through the carbon isotopic composition how high or low carbon dioxide was. The boron isotopes I've already explained to you earlier today, and then there are various other proxies here that are based on leaves. The stomatal frequencies are probably the easiest one to understand. So stomata, these funny shaped cells over here, those are breathing cells. So this is what a tree needs in order to actually let carbon dioxide in. But at the same time, when that tree opens these stomata and lets the carbon dioxide in, it actually also loses moisture. And so the tree has to kind of balance of, how long do I keep these things open and how many of them do I actually want to have? Because yes, I want the CO2, but I also don't want to lose my water. And so it turns out that the more carbon dioxide there is in the atmosphere, the fewer of these cells the tree can afford to have, because with every single breath they take, they get a lot of carbon dioxide in, and they're not losing as much water. There are various different varieties of this proxy. Leaf gas exchange is taking this much further. It also looks into stomata, but looks into models of gas exchange and isotopes paired with those stomatal data. Leaf carbon isotopes are actually very similar in their application compared to what the alkanones are doing, except that it actually averages over a lot of different photosynthesizers. And then the paleosols, they look into minerals that precipitate in soils that also incorporate an isotopic composition based on the gases that are going through the soils. And then narcolite and trona, those are elements that precipitate either above or below a certain carbon dioxide concentration under given temperature conditions. So these are all the different proxies that we looked at, and we got experts from all of these proxies together. And then we had 3 different workshops. One of them, the first one was at Lamont, and then we had another one in Texas and one in California. And so those are just pictures of all the different people that were involved in this. Actually, this is not even all of them, because some of them couldn't make it. And many of them we have collaborated with online. But what we did with these workshops is that, first of all, we decided we wanted to have a better database. So what Dana had done is he basically took the carbon dioxide data and the H data, and he put them together and he plotted them up. It turns out if you really want to go into the proxies and you really want to understand what's behind them and you want to make them useful for the future, you also have to look into all the raw data that have ever been collected and the calibrations and so on. So that would make it a lot easier to recalculate them at some point. And so we created these really elaborate spreadsheets where for every single one of these CO2 estimates you need to enter some 50 data into it. And so it's quite tedious to do that. It took us a long time to do this, to convince everybody to report this information. And in many cases we couldn't even find the raw data. And so that makes it very hard for us to really assess what is going on. Some of the authors have passed away or have left science. And so some of those data are actually lost for us to move forward with. But what you're seeing here is here on the bottom, this is what we call the data archive. So these are all the different studies with all these different proxies that have been published over the last 30 or so years with these different proxies. And you can see that there are some trends. It's a little bit higher over here, but there are also some data that are just super high in the middle. You see that the scale here is actually very different. This is 9,000 parts per million of CO2. These are the data that we then went into, and we had the experts for every single one of these proxies look at and to vet these data to say, okay, we've learned a lot about these proxies over time. We've learned how we have to clean them in order to remove contaminations. We have learned about additional pieces of information that we need in order to calculate them. And so some of the older records are actually not really comparable to what we're doing today anymore. And so based on all of that, we have vetted these records, and we said, we defined which of these records are actually still conforming to our modern proxy understanding. And those are the data that you find here in that top figure. So here you can see that this scale of 9,000 parts per million has now gone down to 3,000 parts per million. You can see a much more coherent picture of the carbon dioxide concentrations, but you also see a lot of problems, and much of the problems come from the fact that most of these records are now based on marine proxies. And that's a bit of a problem, because every single one of these proxies has problems. The isotopic composition of seawater has changed and all kinds of other things. And so this is not actually our end goal of what we want to do with this. We really would like to have all of these different proxies in there, but some of them we actually have to improve. And some of these proxies here, this is what we categorize these data. This is what we call category 2 data. Those are data that are not entirely quantified, where we cannot really quantify the uncertainties yet, because some aspects of this are not known. For instance, with a phytane is actually an element or a molecule that is made by all kinds of different marine algae, and so that makes it a lot more complicated to really say, okay, so what does this mean? Is this a shift in the species that are creating this? You can see that those data, those are the phytane data here. They're quite a bit higher in certain parts of the record than other data. And yet again, others are somewhat difficult to quantify as well. So I'll get back to that in a second. But overall, so these data actually, the gray data on this middle figure, those are the same ones that are here on top. And so overall, there's still a pretty decent agreement between these different estimates. And so they're not bad data. They're just not as quantifiable in the way of how we're treating them right now, how we have treated these data in the past. Now this is where we put this all together. The top figure here, the top blue line, those are the category 1 CO2 data. So time goes from left to right, 65 million years ago to the present. And the top figure, these little bubbles there, those are the actual data. The solid line, that's a correlated random walk model that Gabe Bowen has set up for this, where basically the different time steps inform each other about the carbon dioxide concentrations. And so you get a more statistically robust picture of what the carbon dioxide is doing, except this model is actually binning the data in every 500,000 years. So it's actually, it loses some temporal resolution. Relative to that, in the next panel, that gray line, those are global surface temperature estimates based on benthic foraminifer in the solid line. And then these gray boxes over there, those are surface temperature estimates from surface temperature proxies that have been collected in different time intervals. You can see that all of them are showing a decrease in temperature over the Cenozoic, but they're not exactly agreeing. So there are some differences between them. Now the third panel, that's the sea level curve of Ken Miller, shown in slightly different ways than than shown in the paper. But you can see this big change, for instance, here at the Eocene Oligocene transition 34 million years ago, when Antarctica started glaciating. And these globes here, they're actually showing the North Pole on the upper globe and the South Pole on the lower globe. These are all pictures that Chris Cortese has made for us. And you can see that if you go back to 50 million years, there's no ice on either one of those globes. If you go to 30 million years ago, you see that ice growing on Antarctica, but still nothing in the northern hemisphere. That ice in Antarctica gets thicker and thicker. And then these last globes over here, those indicate the ice extent at the last glacial maximum, when we had this massive ice sheet over the northern hemisphere, big ice sheet over Antarctica. And now the last ones here on the right-hand side, that's just the glaciation of Greenland today and Antarctica today. So all of this is going together. Warmer times, no ice. Colder times, a lot more ice. Sea level is lower as we have more ice on the continents. Here on the lowest picture, this is actually, we were trying to rope in the biologists here and to look into what are the effects of these carbon dioxide concentrations on the living terrestrial environment. And what happened over this time is that we had the shift from C3 plants to C4 grasses. And we could, with these carbon dioxide concentrations, we could now kind of quantify that more to a time that dropped below about 550 parts per million, where that major shift was happening. Of course, all the others, so the evolution of the horses, that's mostly tied to what environment they found. Biology is also very much affected by precipitation, for instance, which through carbon dioxide effects is affected by climate, but there are lots of different processes that are working together in order to create the biological evolution on the planet. So here's our, this is kind of was the print figure that we put into that paper that kind of puts it together again. In terms the colors, they indicate temperature, both temperature and CO2. So both temperature, time, and CO2 are actually shown on log scales. And you can see that during the early Eocene, that's when we had the warmest temperatures and the highest CO2. We have to go back to about 16 million years ago to see carbon dioxide levels that were similar to what we're expecting in the near future. So we actually have to go back 15 million years ago in order to see carbon dioxide levels that were as high as today, sustained for a 500,000 year interval, so not just short blips of that. And then you see this long decline towards the coldest temperatures that we have today. Now here's the last thing that I wanted to show you. So the Cenozoic part is now complete. We now have a follow-up project that looks into this to try and look into all of these other records that have been published already over the last 470 million years or so. The colored symbols here, those are records that we think, based on our modern proxy understanding, that they're still conforming with that understanding, and they're sort of comparable to what we understand today. All of the gray data on here, those are data that are questionable in their current form. So they would need to be modernized, they would need to be updated, where new analyses have to be done for these individual proxy records to really bring them up to the modern proxy understanding. And so that's what we're actually doing with this new project, where we actually have funds in hand to update these samples and to collect more information. So that brings me also to the end. So we've been doing a lot of work with this. This is by no means complete. We're collecting more data. We actually need more data. There's certain time intervals. They have very large uncertainties where we actually have to collect more data because we don't have very many. It's also pretty clear that all of these proxies have problems, and the further back in time we go, the more problems they have. So if we have more of these proxies and they all agree, then we can be more confident that what we're reconstructing actually makes sense. Some of these older, in particular the very old records, they often need to be modernized in order to make them useful today. But we also really wanted to set up this database and bring people in to document their samples in a different way from how we have done it in the past. Very often we went back into these papers and basically only the data have been plotted in a figure, and they weren't even reported in a table, for instance. Many of them never report their original raw measurements and so forth. And so in that regard, all our funding agencies are going after us, and they want us to report these data in a better way. And there's a really good reason why this should be happening. Now in this new initiative that we have, we're also developing more forward proxy system models, where instead of going from the proxy measurement, we go into our calibration and estimate what the condition was until where it was grown. We actually go the other way around. We go from some sort of a preconceived understanding of what the environment could have looked like to model what the proxy value should be, and then we can compare the proxy value to the one that we actually measured, and then learn from that about our proxies, but then also come up with better statistical resolutions of them. So that was the end of, that was the last slide that I wanted to show you. So thank you so much for your attention.
Thank you so much. Jim, do you mind just adjusting the slide? Yeah. Any questions? Let's start.
Yeah. So when it comes to pH reconstructions of extinct species, how do you know the measured values they're actually recording the the pH and it's not offset in the final effect?
That's a really good question. So in the past we didn't. We basically just went for relative changes. And then we've tried to estimate what was happening over that time. We've now, as you know, Lloyd Anderson was actually trying to do this. We've now gone back in time where we look into species that are calibrated today, and then we look at them in the sediment, in the same sediment core, and where we can measure two species at the same time. So we have one that is calibrated and one that is not calibrated. And if they're offset, then we assume that that offset was something that happened in other times as well. Ideally, we'll do that over several different samples, and if we always see that same offset, then we can go further back in time. And so that's a way of how we can cross-calibrate these species against calibrated ones going further back in time. And so then at some point, you have a modern one, you have an extinct one. At some point, you have calibrated that extinct one, and then you're calibrating now that extinct one against another extinct one, and you're tracing that further back in time. Not ideal. But it's an approach to kind of get to that a little bit more closely, because you're absolutely right. There are these differences, these offsets, and so we cannot use the same calibration for every single species.
Yeah.
You mainly focus on tropical species. Is there a reason for that?
The tropical species are more easier. That's mostly for the culturing, but it also, when we're trying to reconstruct climate change or we see carbon dioxide, we try to go into the gyres. Because the gyres are oligotrophic, so there's very little, the carbon dioxide in the atmosphere and the ocean is actually in equilibrium in most of those places. We can't go into an upwelling area. If we go to the high latitudes, we have very different temperature conditions again. We also have very different nutrient concentrations there. So all of these places, if you go into something like what Taro Takahashi has done, these maps of carbon dioxide in the surface ocean of where is carbon dioxide in equilibrium with the atmosphere or not, is mostly in the gyres, and so that's why we're focusing on those areas to do our reconstructions. Of course, sometimes in some studies we're also interested in was there more upwelling, was there less upwelling, and then you would specifically go into those locations. You need different species for that, but that's then a different study. So for trying to reconstruct a global signal, you have to go into these gyres, and that's why we end up with these tropical or subtropical species.
For the PETM, people talk a lot about tipping points in modern climate, and maybe if this is a good analog, it would be interesting to understand whether there are any tipping points that were crossed and if we could actually see that. So I guess I'm wondering whether you have thoughts on that, and also if you have thoughts on why there was no mass extinction at PETM.
So there. The PETM is a difficult one because we have these massive dissolution events, and so even trying to figure out how long did it take to get there is often very difficult because many of these fossils just got dissolved. So we don't really know, where is the tipping point? Where exactly did it happen? Where exactly did species go extinct? Because we often have just a before and an after, and we're missing a big chunk of what was happening at the actual event. So that makes it really quite complicated to do that. In terms of mass extinction, so there were, the PETM, there was some extinction of some species. Most of that extinction was probably mostly happening in the deep ocean, where deoxygenation was a big thing. So benthic foraminifera actually went extinct to very large, very many of those species went extinct. In the plankton, there wasn't that much going on, but there are events even on land that species disappeared and species showed up again. So extinction and evolution was definitely something that was happening then, and that's probably indicating that's what's happening today as well. So that some of these species, like pteropods, for instance, that are pretty vulnerable to ocean acidification, that many of them might actually disappear. We see the same problem with the corals. The corals are also not doing well when it comes to warming. But at the same time, there are also some species that seem to be doing better than others, and so that might be able to survive these events. Life did survive the meteorite impact. It did survive the PETM. So it will also survive now. The question is which organisms will survive and which organisms will not survive, and how many of them will we see as something that might end up on our plates, for instance, as something that we will actually dearly miss. Most of the general public have never heard of the pteropod or foraminifera, and they don't care about them. But when it comes to having a salmon on your plate that may not be able to eat the pteropods anymore, that's all of a sudden when people start feeling it. Yeah.
So on the slide where you had pictures of the plankton, you know, before, during, and after the event, right during the experience dwarfism, and then after they kind of bounce back, have there been any species that you have observed that after bouncing back they evolved to account for ocean acidification, or is there too much variability in the way that the ocean has acidified and then sort of...
You mean were they thicker afterwards?
Right,
Have they actually overcompensated for this? It's not the kind of work that I'm doing. I'm not aware of anybody having seen that they were actually thicker afterwards. I think for plankton it's also, you don't want to be too heavy, because if you're too heavy then you sink a lot faster. So there's probably some other mechanisms that compensate for that. But yeah, I'm not aware of such studies. But it's an interesting question. But then I would imagine that most of that probably would have happened on these shorter time scales of maybe decades to maybe hundreds of years, which is something that is really difficult to get out of the geological record, because you don't have that time resolution in the geological record. Good.
And maybe can
How do you get 15,000 gigatons of carbon?
Where do you get them from?
Yeah, there's only 5,000 in all oil, gas, and coal put together. Estimates of the total lithosphere are 66. So you're releasing 1/4 of the carbon stored in the crust.
So as I said, the uncertainties on that are really quite large, and they're probably larger. So there's follow-up studies that said it was somewhat smaller than that. But most of the studies that we're looking at right now, they're basically saying it's got to be more than gas hydrates. So more than what you can possibly conceive of gas, because gas hydrates, as you know, have this very, very negative isotopic composition. So you can't really do it with methane hydrates. It needs to be something that has a higher isotopic composition than the gas hydrates. And so, I mean, the thermogenic methane sort of, with the quantities that are in there, from what I understand, what the modeling studies are doing, that seems quantitative. It seems doable. But these 14,000, almost 15,000 gigatons, they're on the high end of that. So I think there are other studies that go a little bit lower than that, 10, maybe 7,000 gigatons. It's still quite high. It's still a lot of carbon that goes into it. Richard Zeeby was really upset with us, and he said, "Well, I can't do that with my model. That doesn't fit in there." But we have to use our proxy data to say what it is. I'm not saying those are the right numbers. This is just what our proxies, the way how we understand them, what they can tell us, and they do have massive uncertainties on them. So even for that, we still need more data and different ways of how we can quantify this than how we've done it so far. But most of the estimates that we have so far, they're all coming from modeling, and modeling has mostly looked at methane hydrates. That's mostly what's been coming out of that. And I think the newer evidence is really pointing more to something that has a higher isotopic composition than what methane hydrates have. But I agree with you, these are very, very large numbers. And that's a pretty solid quantity that you need to mobilize. But then also in the North Atlantic, I mean, that we didn't have any deep water formation in the North Atlantic at that time. So this is kind of a place where for tens of millions of years you've mostly deposited organic matter, and you haven't actually washed it out and moved it out with the ocean circulation. So in those relatively organic matter rich sediments, if you burn lava through that and you burn that up, you would probably mobilize quite a lot. But I'm not a modeler, and I don't know what those quantities are. So the newer estimates, there were some recent papers that placed this a little bit lower, not using the boron calcium proxy. It was just one way for us to look at this and to still make sense of these boron calcium measurements that we have taken, to see that these proxies are even working further back in time, even though the uncertainties are very large that we have.
Thank you again.
Thank you.